So, good afternoon, students. And today we will continue with the chapter Sexual Reproduction in Flowering Plants. Till now, students, we have discussed the different parts, microsporogenesis, megasporogenesis, then the gametogenesis we have discussed. Then we have discussed about double fertilization and triple fusion. Now, up to the event of fertilization, see, actually, the reproduction was divided into three events: pre-fertilization, fertilization, and post-fertilization. So two events we have already discussed, that is pre-fertilization and fertilization. And today we will be starting with the post-fertilization events. In the post-fertilization events, there are four major processes. Okay, in the post-fertilization event, there are four major processes. What are those? First, endosperm development. Second, embryo development. Third, ovule into seed development. And the fourth, ovary into fruit. Okay, ovary into fruit. Now, so these are the four post-fertilization events: endosperm development, embryo development, seed formation, and fruit formation. Now, one by one, we have to discuss in detail how this is achieved and what happens. So let us first discuss about endosperm development. Students, endosperm already we have done when we have done the double fertilization. There I have discussed with you all that how the one male gamete fuses with the polar nuclei to form pen. Pen develops into peck, and then the endosperm develops. Can anyone recall what is endosperm? Anyone? So just raise your hand so that I can unmute you. Endosperm. Endosperm. Okay. Before telling the endosperm, can anyone recall what happened during double fertilization? Shweta, unmute yourself.
Yes, sir.
What happens in double fertilization?
In double fertilization, the first pollen fuses with the egg, and the second
1 minute.
may
1 minute. Don't say first pollen. Say first one male gamete. Okay.
yes, sir.
Okay.
And the second pollen fuses with the fused polar nuclei.
Okay, polar nuclei, and this forms the pen, primary endosperm nucleus. Okay, so now you
Yes.
see from here we will connect the double fertilization, and now we will come to endosperm. Now the pen, primary endosperm nucleus, and then the primary endosperm cell, PEC, it divides to form the triploid endosperm tissue. And the endosperm already we discussed in our last class that it is filled with reserve food material, and they are used for the nutrition of the embryo. Okay, so you can see very well in this image, which I have highlighted over here, this part. Yes, these are the antipodal cells. Okay, this is the central cell. This is the egg cell, the synergid cells. Now this is the endosperm area. So this area, the PEC, the primary endosperm cell, proliferate and give nutrition to the developing embryo, because embryo is formed when the male gamete is fused with the egg. Okay, so this is the area where the endosperm proliferation will begin. Now, the next part. Now, for the endosperm development, the primary endosperm nucleus, which is called pen, undergoes a nuclear division to give rise to the free nuclei. And this is called free nuclear endoplasm. endoplasm. Okay, free nuclear endoplasm. You can see over here, the number of free nuclei varies greatly, and the endosperm becomes cellular when the cell wall formation starts. Here you can see that it is free. Okay, but when the cell wall formation start, then it becomes cellular. One example we can have from the coconut, the tender coconut water. Okay, you must all have drink this coconut water. That is a free nuclear endosperm. Okay, thousands of nuclei are there, and the white kernel. Okay, the white kernel is a cellular endosperm. So that is the beginning, the free nuclear endosperm, and then it changes into the cellular endosperm. Okay, so endosperm is clear to all. What is endosperm and how the development occurs in the endosperm? The endosperm is clear. With this image only, we can see how does the endosperm proliferates. Endosperm is clear to all. Through the chat box, you can give the reply. Okay. Okay. I want the reply from all the students. Okay, Khushi Anand. Okay, fine. You asked to explain once again. See. Yes, Khushi. See, the polar nuclei fused with the second male gamete to form pen primary endosperm nucleus. This we have studied in double fertilization. And this pen undergoes a nuclear division to give rise to endosperm. That means it will repeatedly divide, it will proliferate, and it will finally give rise to the endosperm. And this endosperm provide nutrition to the developing embryo. Okay, Khushi? Khushi Anand? Okay, fine. So this was the first post-fertilization event. The second post-fertilization event is embryo development, the most important part. Now the embryo is formed. How the embryo is formed? Niharika, how embryo is formed? How embryo is formed, Niharika? Unmute yourself. How embryo is formed? Yes, Niharika.
Embryo yes, sir. The male gamete which fuses with the egg, after that the embryo is formed.
Very good. When the male gamete fuses with the egg, the embryo forms, and then the embryo will develop into the zygote. So the embryo develops definitely at the micropylar end. Okay, this is the end, the micropylar end. Okay, the micropylar end. And what is the opposite end of the micropyle called? What is the opposite end of the micropyle called? Who knows? Raise the hand. The embryo lies at the micropylar end. Okay, Niharika, very quick. Unmute yourself. Okay, all the students have Chalazal end. Very good, Chalazal end. Good. Very good. Okay, fine. So the embryo develops at the micropylar end where the zygote is present, and this zygote divides after the formation of endosperm, because the endosperm give nourishment to the developing zygote, developing embryo. And now we will study, student, that what is the difference between the embryogeny. Embryogeny means embryonic development in the monocot and the dicots. Okay. Now the dicotyledonous embryo. Now, student, many part of this you have studied in class 11 also. If I highlight this image, which is there on the screen, can you recall class 11? Which chapter you have studied this? The dicot embryo and the monocot embryo? Anyone? In class 11, in which chapter we have studied this? Anyone? Okay, very good. Kushianan, Shweta, morphology of flowering plants. Very good. In the chapter morphology, yes, correct, very good. Now you can see this is a diagram of a typical dicot embryo. The there is at the bottom, there is a root cap. What is the function of the root cap? The root cap protects the radicle. And what is a radical? The radical is your, I'm just writing over here, radical is our future root. Radical is future root. Okay. So this root cap protects the radical. Now the portion, now you can see over here in this image again, these two are the cotyledons. This area are the cotyledons. The cotyledon stores food, and this part is the plumule. And what is the function of the plumule? The plumule is our future. Plumule becomes future shoot. Okay, it becomes future shoot. Okay, and this total area from the plumule to the root cap, this is called embryonal axis. And the portion of the embryonal axis between the point of attachment and the radical is called hypocotyle. Now, if we compare the dicot embryo with a monocot embryo, we can see over here additional structures are named over it, coleorhiza and coleoptile. Coleoptile is a covering over the plumule and coleorhiza is a covering over the radicle. But in case of the monocot embryo, the cotyledon is reduced. And this reduced cotyledon, what is the name of this reduced cotyledon? This is the scutellum. This is called the scutellum. The scutellum is the reduced cotyledon. Okay. So coleoptile is a covering over plumule. Coleorhiza is a covering over radical. This is very important question, student, that what is coleorhiza, coleoptile, and what are their functions? Okay. Is it clear? Is it clear, students, that the anatomy, or we can say the diagrammatic view of the dicot embryo and the monocot embryo? Okay. Okay, Khushi, Shweta, what the remaining students are doing? They are neither responding in the chat box. How much time it takes to write no or yes in the chat box? Okay, fine, yes. Now I'm getting the, okay, fine. Now, we are students, we are coming to, this is again the same part which we have discussed just now. Seed. So students, how many post-fertilization events we have discussed till now? How many post-fertilization events we have discussed till now? Can anyone say how many post-fertilization events we have discussed till now? Good, 2. Fine. Now the third one is seed from ovule. Again, this also we have discussed in class 11 also, the structure of the dicot and the monocot seed. Okay. Let me use me. See here, this is a dicot seed. The outer covering of the seed is the seed coat. Okay. Actually, if we say about, just a moment. Yes, Amardeep, you have joined the class so late. What is the reason, Amardeep? Send me the reason in the chat box that why you have joined the class so late. After 17 minutes, you are joining the class. Amardeep, send me your explanation in the chat box. Okay. Yes, this is the very good, Amardeep. You have given the exact reason. Okay. Okay. And this I will ask you during the offline class. Okay. Okay. Okay, you said you were sleeping. Okay, and then you have said sorry, sir. Okay, try to manage. Okay, try to manage your activities. Okay. Okay. Good to write the genuine reason. Fine. Okay, now Okay. Now, see, this is a dicot seed. And the dicot seed, definitely the seed is a final product of sexual reproduction, ultimately, the fertilized ovule. The outer covering of the seed is the seed coat. And the seed, if we say, consists of three main parts: seed coat, which is a covering, cotyledon, and the embryo axis. The cotyledon stores food. The cotyledon stores food. Okay. Now, regarding the part, we have already discussed what is epicotyle, the portion of the embryonal axis between the portion of the embryonal axis between the point of attachment and the plumule, and the portion of the embryonal axis between the point of attachment and the radicle, that is a hypocotyle. Cotyledon function already we have discussed. Now, students, we will see that how many type of the seeds are there. There are actually two type of seeds. One is a non-albuminous, where there is no endosperm, and it is completely consumed during the embryonic development, like pea, groundnuts. And albuminous, they have the endosperm, like wheat, maize. Okay, so there are two type of seeds. One is the albuminous, another is a non-albuminous. Okay. Okay, students, this part. Yes. Another term, students, sometimes is asked in the examination, that what is perisperm? But in some of the seeds, like the beet or black pepper, there are some part of the nucellus which remain. Can anyone recall what is nucellus? We have discussed already. Nucellus. Anyone? Nucellus. What is nucellus? Okay. Okay. Okay, protective layer. And this remain, and this is called perisperm. This you can see in the diagram, in the image which I have highlighted over here. The endosperm is there. Pericarp is the outer covering of the seed. And this area is the perisperm. This area is the perisperm. Now, next. Now, student, when the seed definitely matures, it dries because there is a reduction in the water content, and definitely the metabolic activity slows down. And that is why we use the word dormancy. And dormancy is a state of inactivity. Can you say, student, which hormone breaks the dormancy? Which hormone breaks dormancy? And dormancy is very much necessary, essential to check that proper growth and development has occurred or not. Zareen, my question, I think you didn't got my question, Zareen. My question was that which hormone breaks dormancy? Hormonal also, the role of the different hormone in promoting the dormancy, in breaking the dormancy, we have studied in class 11 also. That's why I'm asking this part. Can you say? Okay, Shweta. Remaining all the student, answer this question. Yes, gibberellin. Very good. Okay. Yes, now you are correct. Now, what can be the advantage of the seeds? Since pollination, fertilization are independent, seed formation is more dependable because the seed have the better adaptive strategies, and if they are dispersed into the new habitats, and it definitely these are the seeds are ultimately the source of the food reserve. Okay, so these are the source of the food reserves. Advantages of seed, definitely the hard seed could protect the young embryo The embryo is contained within the seeds, and being the product of the sexual reproduction, this is the most important advantage of we can say the seeds. That is, it creates or it generates new genetical combination and variations. Students, you have already studied the chapter Principle of Inheritance and Variation. What is genetic combination? Anyone? Let me see how much you can recall. What is genetic combination? Anyone? Genetic combination? Genetic combination. Okay, let me unmute you. Niharika, what is genetic combination? Niharika, I have given you the option. You can unmute yourself. Okay, Khushi Anand deals with
Yes,
it.
sir.
Yes, Niharika.
Yes, sir.
Genetic combination.
So the combination of male and female gametes.
A combination of male and female gamete is called fertilization. Genetic combination, that means during the time of the fertilization, half of the chromosome is coming from one parent, half of the chromosome is coming from another parent. So the genes, they are combining, new characters are formed, and formation of the new set of the character that creates variation. And variation are the raw materials of, if I give you a fill in the blanks, see, I'm writing a fill in the blanks for you. Let you fill it up. Variation is the raw material for dash. Variation is a raw material of for, yeah, raw material of is the same. Now, who can fill it up? Who can fill it up? This fill in the blank. Since you have studied the chapter Principle of Inheritance and Variation, and I think now you are in the molecular basis of inheritance. Variation is a raw material for, okay, very quickly, let's see. Okay, Khushi Anand first. Unmute yourself.
Yes, sir.
Variation is a raw material for?
Evolution.
Very good. Very good, Khushi. Shweta, is your answer same? You can also unmute yourself.
Yes, sir.
Good. Both of you, very good. Some of you have responded. Okay, Khushi. Okay. Acha, Khushi Kumari 12B, one thing, you are Khushi Ragini or Khushi Kumari? Sorry. Okay, Khushi Kumari. Khushi Kumari roll 5, no? Okay. Acha, 2, both Khushis are there. Okay, fine. Okay. Okay. So, yes, correct. Variation is a raw material for evolution. And that is why this is very important part, because the seeds are creating new genetic combinations and also resulting in variation. Variation also increases the chances of survival of the organism. Okay. Now, the seeds definitely some of the seeds can remain for hundreds of years. Okay. And even that means this can be preserved for future use. The main purpose of this point is that the seed can be preserved for the future use. Now, the fruit from ovary. This is the fourth post-fertilization event we are coming across. The ovary develops into fruit, definitely. And you can see, student, the 3 layers of the fruit. The 3 layers, exo, meso, and endo. Together, they are called pericarp. So the wall of the ovary, that is the wall of the fruit, is called pericarp, and the fruit may be fleshy. Okay, the fruit may be fleshy like guava or dry like groundnuts. There are two types of fruits. False fruit and the true fruit. And if the fruit develops from the ovary, this is called the true fruit. And if the fruit develops from the part other than ovary, okay, like other part of the thymus, then it is called the false fruit. True fruit, like most of the plants, these are the true fruits. If we are talking about the grapes, or we are talking about the mango, but the false fruit like apple, strawberry, cashew nut, these are the different false fruits. Now, students, one very important term is there, parthenocarpic fruit formation. That means formation of seeds, formation of the fruits without fertilization. Banana is the most common example, and even that can be artificially, the fruit formation can be artificially also induced by the application of the growth hormones, and these fruits are definitely the seedless. The parthenocarpic fruit are definitely the seedless one. Now, student, remember, there are two very important terms which need to be discussed because they mimics sexual reproduction. See, these two processes are apomixis and polyimbrine. What is apomixis? Here the seed formation is there without fertilization. For example, like grasses. And always remember, student, that it is a form of asexual reproduction that mimics sexual reproduction. So be very careful. Out of apomixis and polyimbrine, always one question comes in your examination. But don't get confused. Sometimes the student read apomixis is a form of sexual reproduction, but it is asexual, but it copies sexual reproduction. How? The apomictic seeds are developed. Sometimes it happens that, see, this is very, very important. A diploid egg cell is formed without reductional division. If suppose an egg cell, we know the ploidy level of the egg cell. Can anyone say what is the ploidy level of egg cell? Normally, what is the ploidy level of egg cell? What is the ploidy level of egg cell? Normally, Khushi, egg cell, egg will be always, yes, egg will be always, see, no Niharika, egg is N. Egg is N, male gamete is N, they fuse to form 2N. So sometime it happens, a diploid egg cell, that means a 2N egg cell is formed. That means reductional division has not occurred in it. Meiosis has not occurred. And that time what happened, if suppose it is already 2N, so do you expect that the male gamete will come and fuse with this 2N to make your 3N zygote? Is it possible? Okay, yes. No, correct. That is why this 2N will directly develop into embryo without fertilization, because in that case, fertilization is not required because it is already diploid. And we know normally the egg cell is haploid in nature. So haploid egg cell fuses with the haploid male gamete to form a diploid egg. This is a normal diploid. Okay, diploid zygote. This is a normal situation, but here a diploid egg is formed. That means a reductional division has not occurred in the female gamete. Okay, so now it will develop directly, and some species, some nuclear cells surrounding the embryo sac divide and protrude into the embryo sac to form embryo. So both the case, what happened, these are the abnormal conditions. That means a diploid egg is formed and the nuclear cell directly form into the embryo. So this type of situations are called the apomictic seed or apomixis. Apomixis is clear to all? Apomixis is clear to all? That is the formation of definitely the zygote. That is, fertilization is not required. Okay. Now, at last, we will be coming to another special term, polyembryony. Polyembryony, that means occurrence of more than one embryo in a seed. How this can be achieved? Again, the similar conditions, like if there is free nuclear cells surrounding the embryo, then each ovule contain many embryo. One embryo is normal, but when the nuclear cells surrounding the embryo sac divide, then it also give rise to the extra embryo. So students, sometimes this question is asked in the examination, that how polyembryo arises or how apomictic seeds are formed. Okay. With this, students, we have completed the whole lesson. And remember, students, today's notes for the online class, I will be sending you in afternoon or by evening. Though tomorrow you have your exam, no need to hurry up today to write it up. Better tomorrow after the examination, when the examination overs for you, the class test, that tomorrow evening you can write this part, so that this part, the last part of this chapter, will be complete for you. And Monday, when we will have our offline class first, then we will start with the human reproduction. Okay. So now, at the end, let me have an interaction with you all. Any area in this chapter where any problem is there, so that I can clear your doubts? Anywhere you have problem in this chapter? Anywhere in this chapter, you can send your queries in the chat box. Yes, you can send your queries in the chat box. Okay, let me check. Yeah, yes, Amardeep, diagrams are definitely very important. Diagrams are always very important. Okay, Khushi Anand has sent a question. Khushi, it will be ploidy. Ploidy level of MMC, that is microspore mother cell. Any idea, student, what can be the ploidy level of the microspore mother cell? Anyone? Ploidy level of microspore mother cell. Remaining students, you can send your answer in the chat box, so that we will discuss that part. Ploidy level of MMC, that is microspore mother cell. Let me also come to that part. Yes, the answers have come. Okay, Khushi has given the answer. I want this answer, all the students should send in the chat box. All the students. Let me see. No, Shweta. Microspore mother cell, that is a pollen mother cell. Yes, Shivam, correct. Khushi Kumari, correct. Niharika also correct. Yes. Now, Khushi Anand, I'm sharing this answer with you all. That is microspore mother cell or the pollen mother cell is definitely diploid, because it has to undergo the reductional division. I have also shared this slide with you all. You can see over here. The pollen mother cell is diploid. It undergo meiosis to form the tetrad of pollens. Okay, so it is diploid. Okay, Khushi Anand. Khushi, is it clear to you? Okay, fine. Okay. Now, let me ask randomly few questions. Yes. Can anyone tell me what is a function of tapetum? What is a function of tapetum? What is the function of tapetum? Okay, Shweta. Unmute yourself, Shweta.
Yes, sir. To provide nourishment to the growing embryo.
Okay, very good. What next question? What are the two layers of what are the two layers of pollen grain in the structure? Pollen grain. What are the two layers? What are the two layers of? Okay, Kushi, Kumari, unmute yourself.
Exine and intine.
Exine and intine. Okay, fine. Next question is for Shivam. Shivam, unmute yourself. Shivam, unmute yourself. Shivam, why the exine is hard? Why the exine is hard, Shivam? Due to the presence of which chemical? Yes, Shivam. Okay, Khushi, Shivam, please refer and study. Okay, Khushi Anand, unmute yourself.
Exine is hard because of
Oh, Khushi Kumari,
sporopollen.
okay. Sporopollen. Okay, fine. Okay. Okay, students. So our class is going to get over within few seconds. So today's class note I will be sending in the group. Okay. So thank you, everyone. Thank you for the class.